Hi everybody, welcome to another edition of AP Biology. I'm Mr. Minicky, and we're going to spend a little bit of time today talking about the nervous system. You have this information in the note packet that was given to you, and most of that information occurs on page 5 and 6 of your note packet on the human body. So if you want to follow along with some of that information, you can. You can also take out your own separate piece of paper and take down some notes as well to help you along with the nervous system. Nervous system is a very complex system. We're going to pare it down to some of the basic information that you need to know for the AP exam. I've done research in the past on neuroscience, and I could spend days talking about it, but we're not going to do that. We'll try to limit it to some of the most important information, and that involves cell communication. And when we talk about cell communication, we're talking about very short distance cell communication, so that we have cells that are in close proximity to each other but don't actually touch each other in the nervous system. Unlike the immune system, if you remember, those cells actually came in contact with an antigen and then use that antigen to identify self versus non-self. The nervous system's main function is to transmit information from either the outside world, a stimulus, or from the inside world to help you maintain homeostasis. And then your body has to respond to that in a way that maintains that homeostasis. And so the nervous system is really a system that involves control and coordination to help maintain homeostasis. The two major parts of the nervous system are the central nervous system, which is involved in most of the processing and integration of information, making sense what kind of information to pay attention to versus the kind of information to ignore, to habituate to, so that you could ignore, for example, the clothes that you're wearing unless they're uncomfortable, but you should pay attention to something that causes pain, or something that's too bright, or something that's too loud. And so your nervous system then has to control and coordinate and integrate that information into your homeostasis. The organ that's mainly in charge of doing that, of course, is the brain, which is located inside of your skull, and the spinal cord, which is located along the vertebral column. Both of these parts of the body involve paying attention to and integrating the information and making decisions as to what to do. The brain takes a little bit longer to decide what to do. It's involved mostly in senses, memory, thinking, logic, motor control, so in other words, the ability to move. And a lot of that information, in terms of reaction time, how you respond or how quickly you respond to information coming to you from the outside world. Spinal cord is involved mostly with reflexes and also helps to integrate information coming from your peripheral nervous system. A lot of the cells that control this are groups of neurons called ganglia. And ganglia are just groups of neurons that are integrated and work together in order to speed up how fast you can process that information. The peripheral nervous system is the nerves, or the group of nerves, mostly sensory nerves, but also could be motor nerves, that help to control and coordinate what's going on in local areas and organs in your body from the skin or from an organ. And that information is carried along a couple of different pathways. The somatic nervous system, which involves your voluntary skeletal muscles. Those muscles are the muscles that you can think about and move, although sometimes a reflex will cause a response that you don't have to actually think about, just using your spinal cord. And then the autonomic nervous system, a lot of the things you don't have to think about when you think of autonomic, kind of think of automatic. And so, for example, automatic or autonomic nervous system control could be your alimentary canal, your smooth muscle, your digestive system. You don't really have to think about peristalsis and pushing food along, or the secretion of enzymes or bile from your digestive system. Much of that is controlled locally through those peripheral pathways in your autonomic nervous system. It is controlled ultimately by your central nervous system, which helps to integrate and coordinate timing of a lot of these events, but most of that is not conscious control. Your sympathetic nervous system is part of the autonomic nervous system, and so when you think of sympathetic nervous system, you can think of fight or flight, and so during examples or parts of the body that are dealing with stressful situations. During times of stress, you have to be able to mobilize very quickly, and you have to be able to go through the process of energy usage for short-term survival during, let's say, a game, or if you're running away from a predator, or if you're taking an exam or something like that. You're really not thinking about eating. You're really thinking about getting through this. And so during times of stress or anxiety, your sympathetic nervous system is active. If you're really just thinking about, relaxing on the couch and eating something casually, whenever you're just maintaining basic homeostasis, this is your parasympathetic nervous system, and this is kind of during times of quiet. Overall, your nervous system is a very, very quick system, usually occurring over milliseconds of time to respond to some sort of outside stimulus or internal problem like stress. And that very quick, there should be 2 L's there, that very quick response is usually what keeps you alive very quickly to respond to danger. Long range, slow kind of control and coordination system would be more like your endocrine system, which uses hormones to do long range cell communication over long distances. And that is a very slow system that could take hours or days or even months to control and coordinate action. Your nervous system is much faster. These are some of the terms that you were looking up to define the basic terms for the nervous system in general, the control and coordination system for your body. The basic cell unit is called the neuron or the nerve cell. And that basic cellular unit is what actually helps to transmit information in your body, usually electrically, and using neurotransmitters, which are chemical messengers that work over a short distance between nerve cells. The synapse is the gap between neurons. Neurons are not allowed to touch each other. If they did, that could be a real problem with being able to control and coordinate what's known as their action potentials. Their action potentials are the electrical impulse that run down a neuron's axon, or the part of the cell that actually transmits that electrical impulse. The neurotransmitter is the chemical messenger. There are many types of chemical messengers that kind of jump across the synapse in order to either activate or deactivate another nerve cell, in order to excite or inhibit another nerve cell to fire or not fire an electrical impulse. So a lot of times nerve cells will tell other nerve cells to fire another electrical impulse or not to fire. And this is how we control and coordinate our nervous system. Some examples of neurotransmitters, some of the more common ones would be acetylcholine, which is one that's involved with thought and memory. Dopamine, which is involved in the reward system, in our limbic system part of our brain. Serotonin is a good example of one. So are examples like adrenaline, epinephrine, noradrenaline, and GABA, gamma aminobutyric acid, which is an inhibitory neurotransmitter that tells other nerve cells, hey, calm down and don't fire an electrical impulse. You, in your note packet on page 5, have a picture that looks just like this one. This is the picture of a basic neuron, a nerve cell, a part of the nervous system that deals with transmitting electrical impulses. And so this is a cell. It has a lot of surface area. It's one thing that we can notice right away on this cell. Lots and lots of surface area, and most of that surface area you can see is over here where the dendrites are. The dendrites are the receptors that receive information either from the outside world or from other nerve cells. Nerves can actually connect to up to 10,000 other neurons. These cells can make multiple connections, and those connections can either be formed during your lifetime or trimmed, pruned during your lifetime if they're not necessarily needed. And so you don't necessarily always lose nerve cells, but you can lose the connections between nerve cells. You can reinforce connections between nerve cells, and these dendrites help to form connections between nerve cells. The soma is the cell body, which is where most of the organelles like mitochondria, the nucleus, and ribosomes would be found. And then you have what's known as the axon, probably the most or one of the most important parts here. The axon is sort of like the wire, and that allows the nerve cell to transmit an electrical impulse. And the impulse always runs in this direction, in this particular example, from left to right down this nerve cell, from the dendrites to what's known as the axon terminal. Terminal means the end, so the end of the axon. Nerve cells are actually insulated by specialized cells called Schwann cells. These are support cells that actually wrap many times around the nerve cell axon, forming what's called a myelin sheath, a very fatty layer that helps to keep the electrical charge moving down that axon. And what's interesting is the electrical charge only jumps where there's a gap between these Schwann cells, between myelin sheaths, called the node of Ranvier. And so what actually is happening is this particular nerve impulse is propagated or moved in a jumping motion from node to node to node. And this helps to increase how fast the nerve cell can transmit electrical impulse. A cell that doesn't have a myelin sheath on its axon transmits electrical impulses more slowly than cells that are highly myelinated. And so you can have cells whose axons are 1 meter long, but they transmit electrical impulses in milliseconds because they have a lot of this myelin sheath. The more myelin sheath and Schwann cells you have, the faster your nerve cells can transmit electrical impulses.
An impulse is an electrical signal that travels along a neuron. In a resting neuron, one that is not conducting an impulse, the outside of the neuron is positively charged. During an impulse, the outside of the neuron becomes negatively charged. This electrical change travels along the neuron, but it only happens in the nodes of Ranvier, the parts of the neuron that are not covered by the myelin sheath.
Notice that when this electrical charge occurs, the inside of the cell changes its charge. It goes from being negatively charged inside to positively charged. And you may remember from the beginning of the year, we talked about cells being negatively charged because they maintain an electrochemical gradient. And that is maintained, in this case, by the sodium-potassium pump, which we'll talk about in a little bit.
When the impulse reaches a part of the neuron that is covered by a myelin sheath, it jumps to the next node of Ranvier. This makes the signal travel very fast along the length of the neuron.
She's right. And this is called saltatory conduction. Saltatory means to jump. If you know Spanish, then you know the word to jump in Spanish is soltar. And so saltatory conduction is the jumping of this electrical impulse from node to node, which increases how fast that impulse can travel.
As the impulse moves on, the parts of the neuron behind the impulse return to their resting state.
That's important because we don't want the electrical charge to go backwards. It must always go, in this case, from left to right, down the axon from the area where the cell body or soma is, down to the axon terminals. Here you can see the jumping of that impulse till it gets to the axon terminals.
When the impulse reaches the terminal branches at the end of a neuron, it triggers the release of neurotransmitters.
Now, those neurotransmitters are released because when the action potential, that change in electrical charge, where it's normally negative and then becomes positive, causes an influx of calcium ions at the end of that neuron, the axon terminal. So as calcium begins to come into that end, which is this area right over here, this area, the end of the first nerve cell, that absorbs calcium from the surroundings, which is it's liquid around the cell. Remember, a dry cell is a dead cell, so all of these nerve cells are surrounded by interstitial fluid or intercellular fluid. Calcium rushes in and causes these neurotransmitters to be released from vesicles.
Neurotransmitters are chemical signals that diffuse across the synapse and attach to the receptors on the dendrites of the receiving neuron. Receptors are like locks into which only keys that have a specific matching shape can fit.
The distance between this cell over here, the first cell on the top, and the cell on the bottom right is only a few micrometers apart. So that distance allows the neurotransmitters to jump across from one cell to the other and allow the cells to communicate to each other. Imagine again tossing the keys to your friend. You're going to toss them at a very short distance. You're not going to long throw keys to somebody. So if you're doing a short toss to somebody else, that distance allows for quick communication between the nerve cells.
When neurotransmitters attach to receptors, it causes the receiving neuron to begin a new impulse.
Or it could also inhibit that second cell from firing. It could be excitatory, which excites the second cell and makes it then have an electrical impulse, or inhibitory, which tells it don't fire, which is probably the reason why you're not twitching uncontrollably as you're sitting down right now. A lot of inhibitory neurons are controlling your muscle coordination.
There are reuptake carriers in the terminal branches that collect neurotransmitter molecules and return them to the vesicle in the terminal knobs so that the neurotransmitters do not remain in the synapse.
That's very important because what happens is those neurotransmitters are reusable. They're recycled back to the first cell again. And by doing so, it prevents those neurotransmitters from flooding the synapse and constantly stimulating the second nerve cell. Drugs that are taken sometimes cause those neurotransmitters to stay in that synapse and give the person a feeling of being high. In that particular case, it is dangerous because it takes a lot more drugs sometimes to get the same feeling because you're constantly flooding that particular synapse with some neurotransmitter like dopamine or serotonin. That can build what's called tolerance, and that can become very dangerous. So as we watch the action potential jump from node to node, gets to the end to release a neurotransmitter and gets recycled back into the first cell again. And then the information can be transmitted on to another cell. Again, you can have 10,000 or more of these connections between nerve cells, all communicating information and coordinating information between them.
The concentration gradients of sodium and potassium across the membrane of the neuron are important in providing a cell with a form of potential energy. This gradient, and thus the resting potential of the cell, is maintained by a pump in the membrane called the sodium-potassium pump. With each pumping cycle, the pump takes 3 sodium ions from inside the cell. ATP phosphorylates the pump, giving it the energy to change conformation and expel the sodium ions to the outside. The pump then picks up 2 potassium ions, releases its phosphate group, and returns to its original conformation, releasing the potassium into the cell.
So this is important because if we're taking 3 sodiums and pumping them outside the cell, and 2 potassiums and pumping them into the cell, what that does is it causes an imbalance in the charge. This is what causes an electrochemical gradient. And so for every 3 potassiums that are released outside of the cell by the pump, 2 potassiums are coming in. And so this sets up an overall negative charge inside of the cell. It's negative, even though both of these ions are positively charged. It's negative because there's an imbalance. There are 3 positives over here and only 2 positives in here. And so because we've set up an imbalance in charge, the overall charge is at a deficit inside of the cell when a nerve cell is resting. When a nerve cell fires an impulse, everything switches place and more positive charges come in and more negative charges are on the outside. The imbalance switches. If we take a look at the picture on the left side, here we have an unmyelinated axon. This is a nerve cell that does not have, on the top here, does not have myelin, doesn't have Schwann cells. Watch what happens when we send an action potential through here. Sodium and potassium are switching places. The sodium rushes in this case, making it positive, and then it becomes negative behind it. So you have this continuous conduction, but it's very slow. Let's compare what happens in a myelinated cell down on the bottom. Only where there is sodium, potassium exposed to the areas outside of the cell do you actually get a sodium-potassium pump. That node and that node, very fast. Big difference between the speed of those 2 neurons. You can see that in this next picture here. On the left, we have an unmyelinated axon, and that action potential is still traveling. It's so slow. But on the right side, the action potential jumps. This is the saltatory conduction. And saltatory conduction, let me write that down for you. Whoops. Well, it's not letting me write. Here we go. Saltatory. Well, that's supposed to be an R. There we go. Saltatory. Wow. Conduction. You get it. Saltatory conduction. And that's what's happening here. It's jumping. The impulse is jumping from node to node to node. And that speeds up how fast the action potential or change in electrical charge, the electrical impulse, is moving. Obviously, this is a good thing. All right, so let's take a look at what an action potential really looks like. This is a graph of what's known as an action potential. An action potential is a change in the electrical charge that transmits an electrical impulse down the axon of a nerve cell, so you can get information from one cell to the other. All of the things that you think about, your emotions, your thoughts, your plans, your motions that you move when you think about raising your hand, for example. All of that is based on just the movement of salt, sodium and potassium, across a nerve cell, across the axon. The emotions that you feel, the things that you remember, are part of this electrical impulse and neurotransmitter information between nerve cells. And so what you see in this particular graph, the shape that you get, looks very much like for example, a PQRST wave in a heart electrocardiogram. And so if you've taken anatomy physiology, you know that you see this PQRST wave that has this little peak that goes boop, boop, boop. And when there's no more electrical activity in the heart, you get somebody who's just flatlined, boop, right? That's bad. Well, the electrical activity that's being measured in the heart is very similar to the electrical activity that's measured in a nerve cell. You'll notice that the electrical activity of a nerve cell at rest is around negative 60 millivolts, and millivolts is the units that we're using to measure this. At about negative 50 millivolts, you have what's called the threshold. In other words, you have to activate a nerve cell so that it actually causes an impulse to travel once you've breached this negative 50 impulse. Once enough sodium has come into the nerve cell to get that potential from negative 60 to negative 50, slightly more positive. Remember, negative 50 is slightly more positive than negative 60. In order to get the nerve cell to fire completely, that impulse has to reach over negative 50. In other words, you have to stimulate the nerve cell enough to activate the full action potential. And the full action potential is when you get what's called a depolarization at positive 50 millivolts. And so all the way up there, we've got positive 50. This is called depolarization. I don't know if I'm going to fit that, so I'm going to write the rest of it there. Depolarization. Depolarization means the cell is depolarized. You've changed its sign from negative to positive. Let's see what they have to say about this.
An action potential is a rapid change in membrane potential that is governed by the opening and closing of ion channels in the plasma membrane of the neuron. In this tutorial, we will review the events of an action potential measured from a small area of a neuron's membrane. The action potential can be divided into 5 phases: the resting potential, threshold, the rising phase, the falling phase, and the recovery phase. We begin with the resting potential, which is the membrane potential of a neuron at rest. At this point, a small subset of potassium channels are open, permitting potassium ions to enter and exit the cell based on electrochemical forces. Note that there is no net movement of potassium ions. For each potassium ion that leaves the cell, another returns, maintaining the membrane potential at a constant value. As a depolarizing stimulus arrives at our segment of the membrane, a few sodium channels open, permitting sodium ions to enter the neuron. The increase in positive ions inside the cell depolarizes the membrane potential, making it less negative, and brings it closer to the threshold at which an action potential is generated. If the depolarization reaches the threshold potential, additional voltage-gated sodium channels open. As positive sodium ions rush into the cell, the voltage across the membrane rapidly reverses and reaches its most positive value. At the peak of the action potential, 2 processes occur simultaneously. First, many of the voltage-gated sodium channels begin to close. Second, many more potassium channels open, allowing positive charges to leave the cell. This causes the membrane potential to begin to shift back towards the resting membrane potential. As the membrane potential approaches the resting potential, voltage-gated potassium channels are maximally activated and open. The membrane actually repolarizes beyond the resting membrane voltage. This undershoot occurs because more potassium channels are open at this point than during the membrane's resting state, allowing more positively charged potassium ions to leave the cell. The return to steady state continues as the additional potassium channels that open during the action potential now close. The membrane potential is now determined by the subset of potassium channels that are normally open during the membrane's resting state.
Membrane potential refers to the charge across the membrane. In other words, where are all the sodiums and the potassiums? And so when the cell has more sodiums outside the membrane and potassium is inside the membrane, that gives you that imbalance in charge where it's slightly negative inside. That's resting. So resting potential means that the cell is around negative 60 millivolts. When it depolarizes, the cell's charge becomes positive, and that's because more sodiums have rushed in and the potassium has rushed out, and it switches the position of the charge. Repolarization occurs in the falling phase, where the cell starts to become more negative again, and that hopefully becomes your new resting phase, which should be back at negative 60, and it recovers its resting potential. So potential is the charge that exists, and that's caused by the imbalance of sodium and potassium. And action potential is sometimes known as all or nothing. And what that means is either you fire an action potential or you don't. There's no in between. It's not like a nerve cell says, maybe I'll kind of fire an action potential, a half action potential, somewhere around 0 millivolts. No. It's either going to be negative 60 or it's going to be positive 50. And that's it. You either fire an impulse or you don't. And you either have a stimulus strong enough to fire that impulse or you don't. And the threshold is the number at which it becomes a strong enough impulse to fire all or nothing. You have this picture on page 5 as well, and this kind of reviews the information we just discussed. The two cells, this is the presynaptic cell over here, presynaptic cell, or the cell before the synapse. Here's the synapse. And this is known as the postsynaptic cell, or the cell after the synapse. And so what this means is that our electrical impulse is traveling this way in that cell. Then you get your neurotransmitters hopping across from that cell to the receptors on the postsynaptic cell, the signal receiving neuron. And then the new action potential is generated in that same direction down the postsynaptic cell if it's excited, if it's an excitatory neurotransmitter. So here we have a whole mess of action potentials traveling through this cell. I'm going to just zoom in on the synapse, and the synapse is where this blue area is here. And here you can see this really cool animation of neurotransmitters being released by vesicles at the cell membrane, and that's just because the vesicles are made of cell membrane. They can fuse with the cell membrane, and they do exocytosis. Those neurotransmitters land where these little receptors are in the postsynaptic cell. The reuptake occurs at number 5. Reuptake means that the vesicle reabsorbs those neurotransmitters through channels in the cell membrane that allows that to occur. There are many enzymes that are involved in this. Acetylcholine esterase is one of those enzymes that help to increase the speed at which neurotransmitters of acetylcholine are taken into the first neurotransmitter, the first neuron again. And that speed helps you to remember things faster or respond very quickly to different stimulus. One thing that this picture is not showing is the calcium. Calcium usually comes in somewhere over here. I don't know if I can draw that on this. Doesn't look like it. But somewhere around number 1 is where you're going to have calcium that's going to push those vesicles down into the end of the synaptic, the synapse over here at number 3, the terminal bud. One important thing to remember is that when we have a response to a stimulus, that can happen very quickly. Usually the fastest responses to stimuli are called reflex arcs. And a reflex arc occurs using your spinal cord, the spinal ganglia. And so I'm going to take a look on the left side here. First, we have a cross-section of the spinal cord We're looking at a transverse section, a cross section. And the spinal cord is actually in the central canal there. And where it says dorsal root, this is where we have ganglia or bunches of nerve cells. And we're going to pass this person's hand over the flame. And what we're going to notice, I'm going to turn off all of these labels for now. Watch what happens. As she passes her hand over the flame, receptors in her skin sense heat, something hot. This red nerve cell, this is our axon of what's known as a sensory neuron. That sensory neuron, the receptor neuron, sends information directly to the spinal cord to what's called the interneuron or the intermediate relay neuron. They're spelling it differently here because it's British. The interneuron then makes a very quick decision to move the hand away by using what's called the motor neuron. The motor neuron in this case is called an effector. It is in green, and it sends an axon down to the person's muscle in her arm to move the hand away from the flame before she even feels pain. You move your hand away and then feel pain because pain is interpreted by the brain, which takes a lot longer to understand what pain is. But you move your hand away reflexively because that protects you from harming yourself almost immediately. If you notice, there are three synapses involved here. You have the synapse over here between the sensory neuron in red and the intermediate neuron in pink. That's one synapse. The second synapse is here between the intermediate neuron and the motor neuron in green. And the third synapse is between the motor neuron and the muscle cell. So you have 3 neurons and 3 synapses involved in this. A very quick decision is made because your brain doesn't decide anything, just your spinal cord. Watch what happens. There's the sensory neuron. It relays it to the intermediate neuron. There's the second synapse, which then relays the information to the third neuron, which is the motor neuron, which will now move the hand away. That happens within milliseconds. The same thing happens on the right side when you have a reflex on your knee and somebody hits it with a hammer like the doctor does for a reflex hammer during a doctor's visit. There's the sensory neuron sensing the tap of your knee. The intermediate neuron or the interneuron is in purple. That is like a switch that decides to activate the motor neuron in green. The motor neuron sends an action potential down to the muscle. The next synapse then decides, well, it's time to kick the knee, and that's your response to getting hit by a hammer on your patellar tendon, which is under your kneecap. That is your very quick reflex arc.
Somatic reflexes are the rapid, predictable, and automatic responses of skeletal muscle to changes in stimuli. All reflex arcs include 5 functional components: sensory receptor, sensory neuron, integrating center, motor neuron, and effector. Other characteristics, or combination of characteristics, differentiate types of reflex arcs from one another. The simplest of reflex arcs have a single synapse between the sensory neuron and motor neuron. These reflex arcs are referred to as monosynaptic. When an integrating center has one or more association neurons relaying messages to other association or motor neurons, the reflex arc is polysynaptic. An ipsilateral reflex is one that has the sensory neuron entering and the motor neuron exiting on the same side of the integrating center. On the other hand, when the sensory neuron enters the integrating center on one side and the motor neuron exits on the other side, the reflex arc is considered contralateral. When one sensory neuron sends an impulse that activates association neurons in several segments of the spinal cord, the reflex arc is considered intersegmental.
In either case, you can actually count the number of neurons that are involved here. We've got a blue one, we've got a purpley colored one, and then we have two that are kind of red, and there's a third red one up at the top. I mean, you're looking at a maximum of maybe 6 neurons here. Reflexes are very quick because they don't have a lot of synapses. And so the fewer synapses there are, then the faster the information flows between nerve cells. The slowest part of your nervous system is actually where the synapse occurs, where neurotransmitters have to jump across that synapse. That takes time. The myelinated axons occur within milliseconds for information. The neurotransmitters take a little bit longer to jump across the synapse, and that slows things down. This is the reason why it takes a lot of time to make a decision about how you feel about something, or when you have to interpret pain or pleasure, or decide to move, or decide what do you want to eat. Because the number of neurons in your brain is somewhere around 86 billion. And so your brain has tens of thousands of synapses between nerve cells in order to control and coordinate all of these functions between the lobes of your brain. Your brain is made up of segments that loosely have different senses and different functions for different parts of your body. And so, for example, your frontal lobe, which is in the front of the brain, this is the anterior on this side, and this is the posterior or back on that side on the right. Your frontal lobe is involved in a lot of what you do for planning and thinking and motor control and speech, the thinking that you normally think about. Whereas your ability to touch and the map of your body and how you deal with sensations and what you're feeling from the outside world, that's the parietal lobe, and the parietal lobe is up here on top, deals a little bit with taste and a lot with touch and pressure. Your temporal lobe, which is where your temples are on the side, that deals with hearing and facial recognition. Memory as well is in there. A lot of the memories are also interior of our brain, where the hippocampus and the amygdala are. And the hippocampus and the amygdala deal with both memory and fear. Your occipital lobe, which is kind of in the back, and I'm going to use a different color for that one. Your occipital lobe here deals with vision. So it's interesting that we use our eyes, our rods and our cones, which are sensory receptors in our eyes, and the retina of our eyes sends information through what's known as the hypothalamus, the part of the brain that kind of, the thalamus as well, that integrates and understands what's being interpreted and then sends that information to the occipital lobe for us to interpret what we're seeing. For control and coordination, we have the part of the brain that's called the cerebellum. The cerebellum is in the back here. If you remember the family that walks on all fours, they had very small cerebellums, and so they had a problem with balance and coordination. And then finally, the last part that's not labeled on here, but probably the most famous part down here, is called the medulla oblongata. And a part of that is also called the pons, and that helps to deal with a lot of involuntary activities like breathing rate, heart rate, etc. The medulla oblongata actually innervates with a nerve called the vagus nerve, and the vagus nerve controls your diaphragm. It also controls your pacemaker of your heart. And so these are things you don't necessarily have to think about. This is all involuntary. Much of what you're looking at with the brain itself in the cerebrum, all of these other components, the 4 lobes that we see, are part of the cerebrum. And the cerebrum is the thinking part of the brain. A lot of that is voluntary stuff, but not all of it. When you think of the term gray matter versus white matter, here's a nice little comparison. The white matter is interior to your cells of your brain, and the white matter is composed of a lot of cells that have a lot of myelination on the axons. And so there may be fewer cells in the white matter, but there are a lot more myelination to allow for very fast processing. Gray matter is more on the surface of the brain, and those make up more cells. However, they are less myelinated. And so more communication occurs here within the white matter, whereas in the gray matter, it's a lot of processing that occurs and just figuring out what's going on outside the body, for example, or what's going on within the body's peripheral nervous system. You do have some information on the brain on the 6th page, where there's, in your packet, where there's a picture of the action potential, there's a picture of the sodium-potassium pump, and there's also a reflex arc. If you have any questions, please contact me. Good luck with your understanding of the nervous system. If you need to replay this again, feel free to do that. And I will be available for you on Teams, on Remind, and through my email as well. Take care. Goodbye.